I think that what Ed suggested is that I should try to convey a little bit of what we have learned over the years in the medical field or biomedicine. And maybe some of these lessons might be relevant to social sciences. And also there could be bidirectionality in terms of what each field could share with the other. So as Ed mentioned, there's quite a tradition of getting false results in biomedicine. And when 10 years ago, almost 10 years ago, I published that paper, I don't think that I was completely crazy and going out on a limb. There was already empirical evidence to suggest that there were things that were going wrong. And since then, we have accumulated more empirical evidence. We also have more evidence on practices that seem to work. So to try to remedy and to correct my first sin, a couple of months ago, I published another paper in PLOS Medicine on how to make more published research true, trying to put together what we have learned and what are still open questions about research practices that may improve transparency and reproducibility. Biomedical research takes a very long time to accomplish its goals. It can take, on average, 25 years from the first description, first discovery in a wet lab, or currently maybe more in silico or whatever else, until you get an intervention that is tested in a randomized trial or in a clinical study or a big trial testing an effect, a treatment effect, and that study changes the world. In that case, what I'm showing you here is the milestones for the most highly cited study papers for interventions going back to when they started, on average 25 years. And even then, even for the most highly cited papers, the creme de la creme of the biomedical literature, about 35 to 40% of them are refuted within a few years because we perform an even larger and better controlled study. And this doesn't show the effect that we thought that we had identified. So I think it's a major issue of efficiency. How can we improve the efficiency of that process? Make it shorter, maybe cut our losses short if it's interventions or technologies or whatever that don't work, or find those technologies and interventions and methods and whatever else that do work faster. The typical recipe of research practices in biomedicine, I think that this is the same in most of the social sciences, probably even more so, is as I present in this slide. You have a solo siloed investigator that tries to present himself or herself as a principal investigator to gain funding. Unavoidably, since he or she is alone, only small studies are feasible. That also means that one has to be creative about cherry picking one or the best hypothesis among many that may be tested. Much of the work is done post hoc, trying to arrive at a result that would satisfy funders and sponsors and people who are behind the research. And some of them have clear motives, for example, financial or academic sometimes. A p-value of less than 0.05 is enough. Since the time of R.A. Fisher, this hasn't changed in many scientific fields, although the mass of information being collected has grown exponentially. There's no registration, there's no replication, and there's no data sharing because all of those are seen as obstacles in the way of being successful. If you register, then you are cutting your free lunch choices. If you replicate, you run a risk of killing your own self. If you data share, you offer ammunition to your opponents who are competing for the same grants. So when that model operates, empirical studies on fields that have used that model and then had something happen to them, and they adopted replication practice in large scale, have shown that most of the initially claimed statistically significant effects are either entirely false positives or grossly exaggerated. Now, how do we know that? Well, there are some fields that replication can happen and can happen very cheaply. For example, in genomics, the price of running genotyping has gone down about 100 million-fold over a couple of decades. So it costs nothing to perform replication, and actually the current standard is that you just have vast consortia performing genotyping on multiple studies, so discovery and replication is the same. When we do that and we go back to check what we thought that we had found before the advent of these highly intensive reproducibility practices, we found out that the replication rate of the early discoveries early until 2005, and many thousands of papers continue to be published even now, the replication rate was 1.2%. So the success rate of that siloed small sample size, isolated investigator avoiding replication is probably not very far from that 1.2%. There's other areas where replication has become kind of mainstream. For example, for preclinical research, industry doesn't want to waste money, so a number of companies and scientists working on them in Amgen and Bayer have started testing academic research that is proposed to fuel potential for R&D. And when they did that, the replication rate was somewhere between 10 to 25%, even struggling very hard to get the results to work, and even getting the original investigators to participate and share in the replication process. 10 to 25%, pretty low percentage, and as Glenn Begley has put it, the failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong. Some settings in some countries are probably worse than that not so good average. So possibly science in the US has an even worse replication potential compared to other countries. This is a paper that I published with Dan Fanelli, and who knows, maybe it will be refuted soon. But we tried to see whether there's more extreme effects in some countries, and we saw that in the US there's more extreme effects. In particular, when the measurements, the key outcomes involved are behavioral rather than biological, in which case, biology, for example, here we had genetics. Genetics are highly accurately measured currently. The error rate is 0.01%. While behavior is a little bit subjective and open to more noise. So when you have noisy measurements, you get more extreme results in settings that are probably more competitive. And maybe this is why you get these more extreme results. This is a speculation. I'd like to see that replicated. So we're trying to replicate that across different scientific fields and domains, and we'll see what happens. Another dimension that has made a big impact in the biomedical sciences recently is the advent of big data. The amount of information that we can collect is escalating exponentially. We're talking about petabytes being easy to collect. And that means both big opportunities for new discoveries, but also big noise and potentially big error. And typically when you try to combine two pieces of data, one of which is very accurately measured, let's say whole genome sequencing, and the other is very poorly measured, for example, electronic health records, what you get out of the compilation is poorly assessed results, because it's the weaker link that drives the analysis. Now, a number of solutions have also emerged in biomedicine, and I will discuss some of them. So one of them is the advent of large-scale collaboration and adoption of the replication culture. As I showed you, human genome epidemiology was completely transformed from a field that was entirely unreliable, 99% refutation rate, to a field that is highly reliable, 99% replication rate. When we have large coalitions of investigators joining forces, we have consortia with hundreds of investigators joining forces, and they all follow the same principle for designing the study, analyzing the study, and reporting the results. I think that this is ideal when you have a clear plan of what you want to do. If you're really trying to discover the new electricity or the new electromagnetic field that no one has seen, obviously this cannot be done. And it's better for each one of us to try to do it in their own way and see what we can get. But if we know what we want to get, which is 2x2 tables, but just plenty of them, in that case, this is 10 million 2x2 tables that are shown in that slide. We know how to run 2x2 tables, so we'd better join forces. Who's going to do the replication? There's a lot of debate. Could be the same investigators. In the consortium that I showed you, it's just everybody. It could be different investigators of the same school. In some cases, I would argue it's best to have different investigators of competing theories or hypotheses. So Nature and Science occasionally have tried to get competing labs to replicate some high-stake findings to try to validate them. Could be combinations of the above, or if you have raw data and protocols and algorithms and everything available, it could be open to the wide public. I have to say that we don't know the best answer to that. And we're running some simulations with some colleagues from Harvard on models of access to raw data that do become available to a large number of scientists. And I have to say, the simulation so far suggests that it's not so easy to say that, oh, just open up all the data and just have everybody go wild about addressing analysis. Maybe there has to be some structure even on how that is to happen. Registration has been another main theme. It has been pretty successful for clinical trials in the last decade. We've seen recorded information on about 130,000 clinical trials that are in ClinicalTrials.gov, and there's other clinical trial registries as well. The question is, what types of designs could be registered and when? And also, what exactly to register? Should one register the full protocol? Should one register a data set, raw data, analysis codes, everything? There's different levels of registration. I would argue that lots of exploratory research should just not be registered, but simply people should acknowledge that this is completely exploratory. I just tried my best. I data dredged ad infinitum and ad libitum, and this is what it is. It's an interesting result that I found. Just take it for what it is and try to replicate it, and that replication validation effort now needs to be registered. Level 1 is registration of a data set. In lots of observational studies and observational research in biomedicine, we don't really have a protocol. You just wake up at 3 o'clock in the morning, you have a bright idea or a stupid idea, you go to your computer and start running analysis. I would argue that I would like to know what is the potential space of analysis that could be run. So I would like to know that James has a database with 5,000 variables, and Robert has a database with 2 million variables, and Ed has a database of just 3 variables, so that I know what their potential is for running analysis. Level 2 would be registration of a protocol, because there is a protocol and a specific idea. Level 3 would be the registration of the analysis plan, and over the years we have realized that analysis plan and protocol are not the same thing. In biomedicine, I bet it's the same in social sciences. You can have a pretty well spelled out protocol, but the analysis plan could be completely up for grabs and completely modifiable downstream. Registration of the analysis plan and raw data is level 4, and level 5 is open live streaming, where you just make your computer public and whatever you do is visible to the entire community to comment on real time. Sharing data. There's a lot of fields in biomedicine that have struggled on how to share data. There are still not well-answered questions on who, when, and how. This is a list of 20 issues to consider when sharing clinical trials data in particular. And for other types of databases, there's other issues. Some of them may be common, others may be different. I'm very much in favor of sharing data, but one has to be realistic. For example, it's very difficult to unearth some data on past studies. I would think it's more meaningful to focus efforts on having some common rules on how to share data for studies that happen from now on, at least for clinical research. In some cases, there may be a better archiving process. In some fields, this has already happened. So dbGaP, for example, in genetics has already been pretty successful. Databases for microarrays and protocols and data sets like Gene Expression Omnibus are up and running for almost a decade now. But not all fields are at the same level or at the same speed or at the same step. Once you have raw data available, what can you do with that? Well, one first thing is to try to repeat the analysis that has been published already. Most people wouldn't like to do that because they don't have time. But in that case, the editor of Nature Genetics promised to us that if we do it, he will give us a paper in Nature Genetics, which has an impact factor of 40. And we thought that that was really a good idea to put some effort in this. So we got together the best microarray analysts in the world, and we took 18 papers from Nature Genetics. Nature Genetics has a policy for microarray research that raw data, protocols, algorithms, everything, software, has to be publicly available as a prerequisite to publication. So this is the ideal situation. You have everything by default. And out of the 18 papers, only 2 of them could be reproduced. In the cases where that could not happen, the data were not available, the software was homemade and it had disappeared, the methods were unclear, so even experts in the field could not understand what was happening, or the result that was obtained was different. So this is the creme de la creme, probably one of the top impact factor journals, the best scientists, most sophisticated type of research, best tradition of open sharing of everything, and still the repeatability rate was just 2 out of 18. A couple of months ago, we published a paper in JAMA looking at reanalysis of raw data from randomized clinical trials. So randomized clinical trials are like the end of the game. It's the last step in the process of discovery to making sure that something works. Supposedly, they always have a primary outcome. They always have a specific goal. They always have some nice power calculations, well, supposedly. In this case, we tried to see whether there were examples in the literature where people had used the raw data from randomized clinical trials that had been published and tried to repeat analysis on the same outcome, the same primary outcome as was in the original publication. We found 37 such papers, not a lot. I mean, there's hundreds of thousands of clinical trials, but the tradition until now has not been to share that information. And out of these 37, 35% reached a different conclusion on whether patients should be treated or not, or which other patients that are to be treated. Why is that? One might argue that maybe it was the enemies of the investigators who tried to kill their study. Well, actually, most of these reanalyses were done by the very same authors who performed the original analysis. But given the reward system, to publish something, you need to say that you found something different. So it was easier to say that the conclusion is different on the very main conclusion to get that published. What would happen if we were to have hundreds of thousands of clinical trials available with people with very different motives on how to reanalyze the data or use the data or integrate the data or test additional hypotheses? I think that we need to give it quite some thought to try to interpret what we will get out of that openness, which I do endorse. Containing the influence of conflicted stakeholders and authors has also been quite an ordeal in biomedicine. Maybe it's less of an issue in many social sciences, but not all, because I bet that there are social sciences that have strong financial stakes. But there's lots of money in health, especially for drugs, devices, interventions that have manufacturers. So a big question that is currently being debated is who should be the sponsors and the authors of kind of sensitive studies like randomized trials or meta-analysis or cost-effectiveness analysis or guidelines. There's always, almost every month, huge debates about these guidelines being biased because the authors had conflicts of interest with the industry, and proposals that none of the authors of guidelines should have any conflict with industry. And the same thing maybe for cost-effectiveness analysis or meta-analysis or even randomized trials. So these are active debates that are in evolution. This is just showing what's the current status. This is what I call auto-looping, and it's 600 clinical trials done by the 15 most prolific companies in clinical research. If you see a link between the nodes, each node is a company. This means that these are trials that have been co-sponsored and they deal with interventions that two companies have, not just one. If you see an auto loop, the thickness is proportional to trials that only deal with a single company's product. So currently, clinical research is only looking at single company's products. It's sponsored by the industry and is looking at the products of the industry, even though for most treatments we have many options by 3, 5, 10 different companies. We don't know their relative merits because each company is forced, in a way, to generate a research agenda that is tailored to its own products. In the social theories, in the social sciences, the equivalent would be scientists who aggregate around their own theory and don't like to test competitive theories head-to-head in whatever that head-to-head comparison might have been. Better statistics and methods. There have been a lot of efforts in biosciences over the years to improve statistical analysis plan, and I'm not going to steal the thunder from Victoria's talk. Statistical training and improved literacy and numeracy of the scientific workforce, better study designs, introducing some standard features, for example, randomization and blinding of investigators in animal experiments is easy to do, but it doesn't happen about 80 to 90% of the time. And people have also been thinking about checklists. NIH, Francis Collins has proposed that there may be more checklists introduced to try to improve adherence to some key issues for specific types of designs. This is just some empirical data for in vivo studies on some very simple things like blinded assessments of outcome and randomization, which by default could be done in 100% of the studies. These are not humans that you cannot blind. It's animals that you could blind the assessment practically always, and you can always randomize. And nevertheless, it's the exception to have that done. There's lots of biases out there. This is a paradigmatic map of 17 million PubMed papers with mapping 235 biases across biomedicine. I have to say that whenever I try to design a study, I think of a few of them, but I'm sure that many others are escaping my notice. So making people more aware of them and trying to sensitize to the need to control for them would really be essential. Increasingly, we have learned to live with small or tiny effects. So many colleagues feel that it's a pity that probably we will not find another association like smoking and lung cancer. Most of the effect size that we're chasing, especially in fields that are highly prolific like genetics, are shrinking to relative risk or odds ratios of less than 1.05. Currently, it's the norm to have odds ratios less than 1.05. Still, these might be meaningful. And if this is what nature likes to have in terms of the strength of the relationships and associations, who are we to say that we want odds ratios of 5? So this poses an extra layer of demand on the robustness of the design and minimizing error, because even with minimal deviations from a quote-unquote perfect design or a little bit of additional error, an odds ratio of 1.5 could disappear, or an odds ratio of 1.05 could easily be created even if it's null. This is looking at 220,000 clinical trials and 85,000 meta-analysis, and is asking the question of how often do we get medical interventions that have big, very large treatment effects on things like mortality. It doesn't really happen. And we may see lots of small studies with big treatment effects. These are odds ratios, but almost always, whenever we see that, when we run additional studies, the big effects disappear. They either completely become null or very, very much smaller than the original expectation. There's also a lot of interest about formalizing analysis, although I would say that the majority of biomedical researchers are very skeptical about imposing on them a specific mode of analysis for the data. These are clouds of results that you would get if you analyze a very simple association. These are 4 associations of 4 putative risk factors like vitamin D and thyroxine and creatinine and vitamin E with mortality risk. You have the hazard ratio on the horizontal axis and the minus log 10 p-value on the vertical axis, and there's about 1 million points for each cloud. 1 million points because there's 1 million different ways to analyze this data depending on whether you adjust or not adjust for 17 variables. There's many variables that you can think of that could also affect mortality that you may or may not wish to take into account. I think this is very similar to the situations that you would see, for example, in economics, where you also have lots of variables and lots of issues around model specification. How exactly are you going to specify the model? So for vitamin E, for example, which is the example on the right side, depending on what you adjust for, you can make vitamin E be the best thing that you could take, reducing mortality risk, or the worst thing that you could take, increasing mortality risk. You have this U shape, and about 40% of associations in biomedicine are of this type. You can get whatever result you want, provided that you just run the analysis that you wish. There's lots of efforts on improving reporting standards over the last 20 years, starting with Concert in 1996. And the EQUATOR initiative currently has about 200 reporting standards for specific types of designs. Many of them are widely adopted, but not by the majority of journals. The most popular ones, like Concert, probably have become mainstream in the best journals. Prisma on meta-analysis is pretty much the same. Strobe for epidemiology, observational studies, less so. STARD for diagnostic tests maybe is number 4. All the others have been adopted, but to lesser extent. And I think that this is an active struggle to try to get more reporting standards of higher quality also, and with more transparent and evidence-based process in their development to apply across different study designs. And finally, there's a lot of interest about how to optimize reward structures, and whether those changes in the reward structure could also help produce more reproducible research. For example, if we penalize irreproducible research, or we reward reproducible research, maybe it's better compared to if we just reward statistically significant results. This is one such proposal for re-engineering the reward system that I wrote with Moin Khoury a few months ago. We call it the PQRST system. P stands for productivity, which is pretty much what is rewarded currently, but we argue that even for productivity, you could limit it to high impact papers, and maybe also have some productivity linked to publication. So you got funding, did you publish? If not, why should you get funded again? Or you got funding, did you register your protocol? And if you did register your protocol, did you publish within 2 years of completing your study? And if not, again, why should we continue? Q for quality, and fields should decide on what are the key aspects of quality. R for reproducibility, so you get points if research gets reproduced. S for sharing, the more you share, the more points you get. T for translational impact, if you're an applied researcher and you're working on making a difference, did you really make a difference? But obviously these need to be operationalized in each field. And as we try to do that, one has to think of what would be the currency exchange rates. So currently, what's happening is what you see in the first column. We just want to publish. If you publish, you get points. And who cares if you share data? Who cares if you contribute to good quality peer review, or if you educate, you train, all these wonderful initiatives? If you get funded, yes, 5 points per R01. So 5 good papers, 1 R01. Then an assistant professor is 3 points, an associate professor is 10 points, a tenured professor is 20 points. If you get a PhD student, a doctoral student, is 2 points, which means that you can exchange a student for 2 papers, or 2.5 students for an R01. It's like a trade market or slave market. And if you get power, that could be up to 200 points, even if you do nothing. And the question becomes, how should we change that reward system to reach the objectives that we want? So change pattern number 1, which is the next column, says that it does make a difference if your research does get replicated. Even better if it gets translated. If it gets refuted, you lose points. If you share, if you contribute to review, if you educate, you get points. And lobbying has no value. And the more radical change 2 column shows a system where not only that happens, but you have no ranks, so everybody's equal. And if you gain power, you become a leader, dean, president, provost, whatever, you lose points. And if you get grants, you lose points, because you need to deliver. I mean, you got all that money. What did you do with that? There could be collateral damages with this, obviously, because then nobody would want to become a leader and nobody would want to get a grant. But some balancing may need to happen to realize which is the best solution. And finally, biomedical research is heavily embedded within a universe of many stakeholders, and some of the social sciences probably have the same issues. So in biomedicine, you don't have just scientists sitting in their computer or in their desk. You have the industry, sales, marketing, R&D. You have private investors, public funders, some of them open, others closed, like the military. You have non-for-profit funders and philanthropists. You have journal editors, for-profit publishers, professional scientific societies, universities, non-for-profit research institutions, supporting staff that are struggling to survive, and if you don't get a grant, they would need to be fired, hospitals and other professional facilities offering services that they want to make money, insurance companies that try to set contracts with hospitals and with physicians and with services, and it's a crazy world, and then government trying to invade that space or remove itself from that space, and state authorities and federal authorities. and common people who are treated or get preventive interventions and so forth. And these people have very different perspectives about what is important for them, for science. Is it publishable research that they mind about? Is it fundable research? Is it translatable research? Or is it profitable research? So getting all of these stakeholders to agree and make sure that we get the best eventually in terms of credibility and reproducibility is not very easy. To conclude, by now we have very good evidence that many biomedical fields have met with very high rates of non-replication, sometimes getting very close to 100%, fields that could just be thrown to the wastebasket. Research practices remain heterogeneous across different fields. Some fields have adopted practices that have led to substantial improvements in replication rates and transparency, but again, this is also very heterogeneous. And I think that we need more empirical research because research on research practice is very limited or even altogether missing in many fields. So getting these gaps filled could really make a difference. I'm closing with a slide of some of the 3,000 people who have been unfortunate enough to collaborate with me in different papers. If anything that I showed you was wrong, it's my fault. What they did was perfectly fine, and I'm grateful to them. Thank you.